translational. So there are multiple approaches when we talk of targeted. So there is one thing that we know as the selective reaction monitoring. You have multiple reaction monitoring, so SRM and MRM. Also you have parallel reaction monitoring, that is PRM. You have SWOT MS based techniques, that is again a DIA approach, but as I said, multiple instruments have multiple specifications, and the work, your workflow will also be quite influenced by the instrument you're working with. And what do you do? So Skyline is a very famous software tool that we would briefly discuss. We don't have opportunity to go deeper into it, but maybe in some next GIAN course we can discuss Skyline in detail. So SRM is the work on the selected ion chromatograms. PRM works on the extracted ion chromatograms. So does DIA SWOT, and DDA is for the extracted ion chromatograms of MS1. So we talk of the word tandem MS, MS. So MS1 extracted ion chromatograms are. So what do you do is you take the DDA, LC-MS primary data, you make a spectral library. What is a spectral library? It primarily is tells that these are the spectra, these are the peptides that could be detected in your sample. And remember, not all peptides will be detected by the instrument. For a given protein, not every peptide, and you know the reasons we discussed yesterday why we cannot detect all the peptides, right? So much of our studies are basically, what is the length that we need to have? Between 7 and? You have exams today, so you should have studied harder. So 35, 7 and 35. So, so you do a selection of target peptides, then you go to the SRM and PRM methods, you set up the method where you just detect only those specific 8 to 10 product ions, right? So now you are cutting down the job for the instrument. The instrument is not looking for the entire peptides, all the ions. You are selectively telling the instrument to look for only those guys with red hats, right? If I put you up in a traffic junction and tell how many people are or how many cars are passing by, it will be a very difficult job for you. But if I give you a job that, okay, count the number of yellow cars, that immediately the load on your cerebral cortex will significantly come down. Or maybe I just tell about count the number of red hatchbacks or red sedans. Then again, so this is in very dumbed down format what a targeted proteomics approach tells. So we, using the software, tell Skyline, and we do a method optimization, and we select only 3 to 5 ions, right? We select very less targets, and this makes it very precise. The instrument is only looking for that specific signal that should come from the sample. So of course, this is the workflow for a targeted. So you know, you do proteolysis, you do peptide separation, you do electrospray ionization. Then what you do is, this is a typical exposition of a, do you have the laser pointer, volunteers? Anurag, the laser pointer. So what you see, Q1 is the mass filter. Now see, here I have put a mass of 0.7 m by z. There are multiple balls, right? You have red, orange, green, blue, purple. But what I am selecting is only the balls that are green. That has a charge by mass ratio of 0.7. Mass by charge ratio of 0.7. What I do in the next quadrupole, it's a collision cell. So here we do some HCDs, high energy dissociations, but these peptide fragments are broken down further and it generates Y ions and B ions. Now this Q3 is again a mass filter. This is a triple quadrupole, right? Q, Q, Q. So this is a quadrupole, this is a quadrupole, this is a quadrupole. So in this final quadrupole, since I have selected only 0.7 m by z, only these specific dots are detected. So it lowers the computational load And these collision cells and the mass filters, so we have two mass filters and one collision cell, so they specifically help you into a very targeted approach. And you see, of all these precursor ions that we got, we are only looking for this trace fragment ion. So this is why we call it the selected reaction monitoring, right? So that's the reason. So if you again go back to the terms, we have SRMs, PRM. We'll come to PRM very soon. Now, what if during the SRM run, what happens is MS continuously acquires spectra at the expected M by Z, right? And precursor ions are selected from Q1, fragmented in Q2, and target specific fragment ions are selected in Q3 part. So this is the basic workflow. I think it is clear to you. We will discuss further when we have more time. So again, selected reaction monitoring. So again, the same things. So you have mass filters that will select for you, right? So you are standing at a traffic junction, right? To make things easy for you. So suppose you are three friends, right? So you are standing at a traffic junction. Your first friend will allow only allow all the cars to come in. The next friend, for better color detection, is standing with a jet spray of water. So all cars that pass through the Q2, they are sprayed. Now you are only standing at Q3. This is another mass filter. So now you can very specifically detect that how many red cars passed through. There is no issues. Maybe there's enough light for you and you can give a good detection. This is a very analogy that you can think of that we do in targeted proteomics experiment. Now, if you don't want to use a mass filter like triple quad, if you want to use an orbit trap mass analyzer, what you have to do is you can use something known as the parallel reaction monitoring. So this is again almost a similar approach, but here our mass analyzer is an orbit trap. It's not a triple quadruple. So you have a precursor ion. There you have a mass filter. It's like a quadrupole. Then you have a collision cell. You are breaking down these same ions, and in the orbit trap mass filter, you are detecting the specific ions that you need to look into. Is the video on? YouTube links are on? YouTube. YouTube. WhatsApp. So PRM is also referred to HR, high resolution SRM, or HR stands for high resolution SRM or MRM, because it involves the use of high resolution mass spec, especially orbit trap and TOF analyzers. So the same technology, the fundamental remains the same. When you upgrade the technology, you can and use an orbit trap or a TOF analyzer, and it exploits the trapping ability of instrument for better resolution and increased sensitivity. So the idea of a targeted experiment is that you have a very predefined hypothesis that I want to detect a specific particular parasitic antigen in the blood of my patient, or a specific protein that is known to increase the predisposition for cancer in the patient plasma. So that makes it very easy for the instrument. And since you already have, once you start understanding Skyline, although this is not part of the syllabi, all of you can look up on Skyline, and there are very good YouTube tutorials by Brendan. So you can look up Skyline, and they have a very good, their YouTube channel is filled with tutorials. And if you're only into targeted experimentation, because most of us don't actually go till targeted. So what PRM does, it reduces the assay development time, it eliminates most interferences and enables confident confirmation of peptide identity, because you are doing multiple reaction, you are doing parallelly SRM, so it's more like high resolution SRM, So there it reduces the assay development time. So PRM developments, so SRM, it takes about maybe 15-20 days in a very well-established lab. It will take over a month to develop, because you will go through the entire samples. You have to create the lower limit and upper limit. You have to quantitate with labeled peptides, all these heavy labeled peptides. So you have to do that. PRM significantly, because it has a higher resolution and sensitivity, can reduce your workload. So what usually people do, this is by Brendan. So what people usually do is you can use servers like Panorama, and again, this is an ecosystem, Skyline, Panorama, where you generate your own data, you upload to Panorama, and they, based on your instrumentation, will suggest, okay, these are the peptides these instruments can detect properly. So since we are not going deeper into it, I'm just giving you a brief. So what is the ecosystem for, and these are all open source softwares. If you want to learn, you can learn it from your home. They have enough tutorials, and they have also some data sets that you can use to train yourself. So basically, I will just tell you now how to perform a targeted proteomics experiment. So there are a few methods by which you can make it. So first is the RT libraries. What is RT? Retention time. So since we're even a triple quadrupole will work on the principle of chromatography, and we'll have something known as the retention time. Then you have the spectral libraries, the spectrum that it has been generated for each of your sample. Then you can use methods and previous measurements, and you do a lot of targeted method refinement. So this is where you spend most of the time, because in your first discovery data, so you usually start with the discovery data. The spectral library is generated from a discovery set data that you have done this entire sample, and in this entire sample for one specific protein you could detect, suppose, 75 peptides, right? And out of these 75 peptides on a triple quadrupole, all 75 might not be detected, right? So you will take the spectral library of 75 peptides, you will use the retention time libraries, and then you will do a targeted method refinement. Out of these 75, you might come down to only 3 and 4 peptides. But these peptides are very robust. Every time you inject the sample, it will detect at a very low limit of quantity. You can also quantitate how much of your target protein is present by creating this simple standard curve that you do in protein quantification, right? So you can do quantification, you can do very sensitive detection. And how do you do it? So you have protein targets, and that you can generate from the peptide atlas, the UniProt server, from KEGG, and you create, for these targets you have peptides. As we discussed, for one protein you have multiple peptides. These peptides are what? Made out of trypsin proteolysis. Then you create the transitions. Transitions means how each peptide is broken down further in the dissociation cell, what kind of Y ions and B ions they produce. Then you do the LODs, the LOQ, the calibration curve. Then you create a linear range. So this is more of a quantitative profiling. Then you try to assure the specificity, accuracy, and precision. And then you also design the assay range and robustness. How much of the sample you need, how strong your assay is. Like, is it working on all sample types? Is it working on only specific sample types, right? So suppose you're talking of detecting viral peptides. Now, when the pandemic came, we came to know that even lions and tigers and monkeys are getting infected. Even minks, all mammals kind of became reservoirs for the virus. Suppose you're trying to look at a pest, something that is zoonotic in origin, and you have a reservoir host and you have your patient. Now, can you detect in both? Can you take a bat blood and detect the peptide, or is it working only with human blood? So all these kinds of assays, because when you develop an assay, it has to be robust. It should work each time, every time, and it should work with precision. So This is primarily from this paper. You can look at it. So when you design a targeted proteomics experiment, you have, so this protein, you see protein 1, protein 2, these proteins are not generating peptides from every fragment, right? So you have specific peptides. You select these specific transitions. So look at the yellow colors. So these are the transitions from the mass spectra that you are selecting. And then you optimize what is the fragment, what is the retention time, and what kind of data can you generate for each of these specific peptides. So one protein can generate multiple peptides, and from out of those multiple peptides, all you need to do for a robust data is about 3 to 5 peptides. If you can analyze and make your assay very robust, you can take it to the clinics. So this is just, I will go through this in very briefly. So you start with a protein sequence search. I think all of you know this is a database, is UniProt. Then suppose you search for P53, which is a cancer specific tumor antigen. Then you upload it on Skyline. Now Skyline, based on the protein sequence and based on the enzyme that you will add on the database, from the database, you know the protein primary sequence is there, right? And Skyline, you can computationally detect that, okay, these are the peptides that can be formed. Next, they will create these kind of precursor ions. So you have Y2, Y3, Y4, Y5, Y6, Y10, till these are the ions. We'll discuss about these ions sometime very soon. So you see, this is the fragmentation pattern. So these are the Y ions, these are the B ions. It's just when you cut the NH2 side is the B ion. So for this specific fragment of peptide, if the dissociation is here, the B1 will carry F and the Y11 will carry the rest, right? So this is the basic schema of Y ion and B ion formation. This happens in the collision cell, right? So high energy dissociation, so this dissociation just breaks up the peptides. So you see, out of all these, you have multiple factors like what is the M by Z at Q1, what is the M by Z at Q3. Q1 is the first quadrupole, the mass filter, Q3 is where we are detecting it. Then you have the fragments, so you can detect Y5, Y7, Y8, Y10, and these are the dwell times. So dwell time is in that trap. How much time is that ion particularly spending? So it's 250 milliseconds. So it's a very short span of time, and this entire acquisition, so accumulation and acquisition time is 250 milliseconds for each of these sequences. So if you add it up, that becomes the cycle time. Again, you have to number of refinements that you have to make sure. What is the abundance? We discussed, right? Is your peptide or protein highly abundant or is it low abundant? What is the measurability? Can you measure the peptide? I mean, not all peptides will fly. Is it prone to degradation? Trypsin. Are you using trypsin? Sometimes you can't use trypsin for specific reasons. So that will scheduling, optimization. So there are multiple ways by which you can do a targeted method. Refinement, it's a long process. Discovery is quite easy. You go till trypsin digestion, you clean up the peptides, and then you allow mass spec to do all the stream. When you want to do a targeted approach, these targeted approach are fairly difficult. The reason they are difficult is because now you have to refine and you have to specify that this is the peptide, but since they are also difficult, they are very precise because you have spent a lot of time optimizing. So these are the outputs of Skyline. So if you can see, these are the specific ions. Is it visible to all of you? So this is the first one is Y7, then Y6, then Y5, Y4, Y3, Y5. So this is the specific peptide, and these are the retention times that could be, these are the specific ions, and this is the charge on the peptide. So this is the result output, and this is what a selected reaction monitoring looks like. So for one specific peptide, you get very sharp peaks like that. And if you're getting such peaks, that means you have created a very robust assay. So the robustness of your assay is shown by these peaks. And again, one more thing that you need to think of it, because since you already have your computational data that is coming from the Skyline database, right? Skyline will predict what could be its retention time, what would be the data you are achieving. Then there is an experimental data, which of course will be different from what we have already achieved. You might have, those who are into experiments would realize that whatever is written in the books is fairly different from what you get. So that gives us the dot p value. So dot product value, so dot p or dot product value, a perfect is 1, is a perfect correlation. Whatever Skyline predicted, you got. You have a value of 1. But the range is from 0 to 1, and just like correlation, anything less than 0.8 is fishy, anything less than 0.5 is extremely doubtful. So, but again, a lot will depend on you as an experimenter, that what you set up as the baseline. If you're trying to detect something, maybe in animal biology, you can go for some lower limits. But suppose your decision means life and death for a patient. Your decision means a patient might get an ICU bed or might not get an ICU bed, right? You have taken up a prognostic tool and you are telling that this specific person might get into severe dengue in the next 24 hours. That could mean a life and death. So there you have to really have a very high dot p value, so that we'll try to achieve 1. 1 is a perfect score. Nobody achieves that. But you see, these are 0.99. So these are actually, you can go till 0.99. This is 0.82. So these are specific dot p value for this target protein RAS2. This is again the MS spectra that is showing the same. So I think I will end here, because the rest part is not very much necessary for you, but I would love to show. This is something that we did at IIT Bombay along with, this is Professor Sanjeeva's slides. So we wanted to see that if we can detect COVID, the SARS-CoV-2 viral peptide in nasal swabs, right? Because they're already mass spec. So you might ask, there were already PCR based studies, but PCR later on we understood gives us a lot of false positive results, right? Proteins are much more robust in terms of detection. So what we did was we took some positive samples and we tried to develop an SRM assay, and here we could detect the SARS-CoV-2 nucleoprotein and SARS-CoV-2 glycoprotein in the samples. So of course, because there is a lot of work to do as far as infrastructure development in hospitals are concerned, and mass specs are still very, mass specs are extremely very, I would say a costly instrument to handle and costly instrument to maintain. So there is some issue with translation to clinics, but actually if you go to labs, tests are ready. Maybe we need a more cheaper intervention, more cheaper or more scale development. Sometimes, like real-time PCR was a real novelty back in 2005, 2006. Even till 2018, it was a novelty, or until the COVID pandemic happened. And suddenly there was so much production of real-time PCR instruments that now real-time PCR instruments are really cheap. So I think I will stop here, and we can, where is the webcasting team? You can pause the recording. So if you have few questions, we can take it. Maybe I will take one.
Just work with the input file.
अभी आ रहा है. For making input file, first we will copy all the accession ID. This is accession ID, and paste in another Excel sheet, and similarly copy all the samples from sample 1 to, I have 12 sample, so from sample 1 to sample 12, copy and just paste corresponding to the accession ID, and then like this is accession ID, and all this I have all labeled. After that, control 1, control 2, so this is the metabolist input file. Here column 1, this is the accession ID, and this is the LFQ intensity of the control and MOHE samples. MOHE, you may consider it as a disease condition. I have worked on hepatic encephalopathy, where I have used moderate grade hepatic encephalopathy, so you can consider it as any disease condition.
You have 6 controls and
6 control and 6 disease condition. And in place of blank, if there is other letter, other colors like NaN, then you will have to remove, or 0, you will have to make it blank. You can see samples are in column, so for that we will use samples in column and unpaired. You know about paired and unpaired samples. अनपेयर्ड यानी कि एक सैंपल अगर सेपरेट है हमारा और एक का दूसरे पे इफेक्ट नहीं पड़ता है तो उसको हम अनपेयर्ड बोलेंगे. अगर हमारे पास ऐसा डेटा हो जिसमें इसका जो वैल्यू है इसके बेसिस पे इस दूसरे वैल्यू में अगर चेंजेस आ रहे हो तो उसको हम पेयर्ड बोलेंगे. हाँ, लेकिन हमारा यहाँ पे ऐसा नहीं है, सब इंडिविजुअल सैंपल्स हैं, सो यहाँ पे हम अनपेयर्ड यूज करेंगे. तो सैंपल इन कॉलम्स एंड अनपेयर्ड, एंड फ्रॉम हियर चूज फाइल वी विल सेलेक्ट द इनपुट फाइल. Submit. Can I submit? So this is the data summary. Here you can see there are two groups were detected in the samples, which you can look back into the input file. Control group and other MOHE group, which is disease group. Also 104 proteins features, which is in our case proteins, with constant or single value across all the samples were found and deleted. And we have in our data there is 6% missing value. So before processing to the statistical analysis, firstly we will have to remove this missing value. And how we will do it? Select missing value, and this is the parameter which we can set according to our need. This is 50% missing value. So generally it is recommended to use more than 30% missing value. More than 30% missing value, it means if any protein in a sample, if it is present at least in 70%, then that feature will be selected, otherwise that will not be selected and it will be deleted from the data set. So we will be using 30% of missing value threshold. And for missing value imputation, there are various parameters which you can use.
Missing value to 30%? Missing value में वहाँ पे 30 सेट करेगा।
हाँ 30%.
What exactly is this missing value indicating?
Here, this is the accession ID of a protein. Here you can see missing value. Here you get the value, but in these samples there is no value. So this is the missing value. So if in at least 70% sample, if any protein is identified, then we will incorporate in data. Otherwise, we will remove that features. Okay. And for missing value imputation, these are the parameters which we can use, and this is the default. If you don't know anything which features will be better for us, then you can use this first one. For my study, I have used estimating missing value using GIAN sample wise. सैंपल वाइज यानी कि एक नियरेस्ट नेबर सिंगल एम्पुटेशन मेथड है, और इसको हम सैंपल का जो मिसिंग वैल्यू है इसको इम्प्यूट करने के लिए यूज़ किया जाता है इसमें। और इसका एल्गोरिदम के बारे में तो मुझे डिटेल नहीं मालूम है, लेकिन ये तो अगर हम सैंपल वाइज अगर सेलेक्ट हम कर रहे हैं सैंपल वाइज अगर हम सेलेक्ट करते हैं इसको, तो जो हमारा ये स्टार्टिंग का 6 कंट्रोल है और उसके बाद जो 6 है वो डिजीज़ कंडीशन है। तो जो स्टार्टिंग के 6 हमारे एक ग्रुप के हैं, इनका जो सैंपल वाइज वैल्यू है एक प्रोटीन का, उसके अकॉर्डिंग ये आपका ये मिसिंग वैल्यू को इम्प्यूट करेगा। Next we will do data filtering. You can simply read this. For data filtering based on the last two categories, the default parameters follow the empirical rule. First, less than 250 variables, 5% will be filtered. Between 250 to 500 variables, 10% will be filtered. Between 500 to 1000 variables, 25% will be filtered, and over 1000 variables, 40% will be filtered. You can turn off the filtering by dragging the slider to adjust the percentage to the filter out when to be 0 when your data contain less than 5000 features, to control the computing time over the server. It means अगर हमारे पास 5000 फीचर्स से 5000 फीचर्स अगर हमारे पास कम अगर डेटा हमारे पास है, यानी फीचर्स यानी कि हमारे केस में क्या है, प्रोटीन्स और पेप्टाइड, अगर 5000 से अगर कम है तो जो यहाँ पे एक परसेंटेज है दिया है फिल्टर आउट करने के लिए, इसको हम 0 पे सेट कर देंगे, जिससे जो कंप्यूटिंग टाइम है वो कम हो जाए. इसको यहाँ से हम ड्रैग करके 0 पे कर देंगे एंड देन सबमिट. And after submit, we will hit on proceed button, and then we will do normalization. For normalization, we can use sample specific normalization, normalization by sum, normalization by median, and other also. And I have used normalization by median. And then data should be transformed into log. Why data is transferred into log? Okay. जैसे कि एक सिंपल, अगर हमारे पास 2 सैंपल है, उसका एक का एक्सप्रेशन सपोज करिए 1 है और एक का 1000 है. अगर इसको हमें एक ग्राफ में प्लॉट करना हो तो हम इसको नहीं कर पाएंगे, क्योंकि एक वैल्यू बहुत ज़्यादा है और एक वैल्यू बहुत कम है. तो इसीलिए इसको हम लॉग में ट्रांसफॉर्म कर देंगे तो इसको हम ग्राफ में प्रेजेंट कर पाएंगे, इसीलिए इसको हम लॉग में ट्रांसफॉर्म करेंगे. And after normalization, you can see the results. This is before normalization and this is after normalization. If for performing statistical analysis, data should be in a normalization Gaussian distribution curve. So this is the proper Gaussian distribution curve, but here it is not a bell-shaped curve. Now data has been normalized, and then we can do other statistical analysis. Whole chain analysis. यहाँ से हम जो यहाँ पे जो रेशियो दे रखा है, इसको हम यहाँ से हमें चेंज करना होता है. हम किसके रिस्पेक्ट में हम स्टडी कर रहे हैं उसके अकॉर्डिंग हम यहाँ पे सेलेक्ट करेंगे. हमारे केस में क्या है, हम कंट्रोल के रिस्पेक्ट में डिजीज़ कंडीशन में क्या चेंजेस आ रहा है उसको हम देखना चाह रहे हैं. तो उसके लिए हम जो सेकंड ऑप्शन है जिसमें कंट्रोल डिनोमिनेटर है, उस ऑप्शन को हम सेलेक्ट करेंगे। जिसके रिस्पेक्ट में आपको चेक करना है वो आपका डिनोमिनेटर रहेगा। बैक
ग्राफ के बाद
क्या हाँ हाँ बैक जाने पूरा दोबारा करना पड़ जाएगा। हाँ स्टार्टिंग से ठीक। Peak intensity, sample in column, unpaired, then choose input file. Here you can see the data processing summary. Here we have two groups, so here two groups are clearly identified. If you are analyzing and you have two groups and you are not seeing here two groups, then there is a mistake in input file. So firstly you will have to correct those mistakes, and then there is 6% of missing value. So firstly we will do missing value imputation.
नंबर ऑफ फीचर्स हमने लिखा था बाद में जिसको 0% किया था, तो वो हमें कैसे पता चला था कि 5000 है, क्योंकि इस वाले डेटा में तो
ठीक ठीक बता देना। Here you see in column, in row 1 and 2 there is no protein, and just go at the last. So this is 2174, 2174 and 2 माइनस इसमें सम कर देंगे, 2172 प्रोटीन्स इसमें आइडेंटिफाई हैं। ठीक है, तो ये 5000 फीचर से कम है, इसीलिए यहाँ पे हम डेटा फिल्टरिंग जो हम कर रहे हैं वहाँ पे 0 पे करेंगे। और यहाँ पे मिसिंग वैल्यू ये हमारे ऊपर हम कितना इसको सेट करना चाहते हैं, लेकिन रिकमेंडेड ये है कि हम मिनिमम 30% को हम सेलेक्ट करें। हाँ बता रहे हैं, आ रहे हैं उस पर। GIAN sample wise. Normalization by median, log transformation. स्लो करें, ठीक, कहाँ तक एक बार। After selecting normalization by median, log transformation, and in data scaling, you can we have selected none, and then submit. And without selecting this normalization button, if you will go to proceed button, then this settings will not apply. So before tapping proceed button, firstly tap after selecting the parameters, select normalize, and then you can view the results. This is showing the sample wise. You can also using this icon. You can also save this. You can also using this icon. You can also save this. Yes. Yes. Decrease the number, and for doing that you will have to increase the whole chain threshold. So and here direction of comparison would be Yamaguchi disease versus control, and then this whole chain analysis is before performing any statistical analysis. So for our study we will not be using this result. This is for general observation of the data. And again, using this button you can save the image. Now we will do t-test. If I have two sample as two group, and if you have three group or more sample as two group, and if you have three group or more group, then you then for that so here only two group, then we will do t-test. And you can just read for large data set more than 1000 variables. We have 2172 variables. So and the group variance will be ignored, and the default parameters will be used for t-test to save the computational time. If you have choose non-parametric test, then Wilcox rank sum test, the group variance will be ignored. So we will be doing unpaired t-test, and t values should be 0.05, and we will select here not FDR, we will select raw. And then submit. Raw is the LFQ intensity. Fast discovery rate. So here you can see in t-test we have identified 1802 है, अगर हम इसको 1.25 रखेंगे, 2 है, अगर हम इसको 1.25 रखेंगे, then when I have used full chain 1.25, then we get significantly down.
डाउनलोड करना है।
हाँ, उस पर इस वाले बटन पे हम सबमिट क्लिक करेंगे और सेव कर देंगे फाइल को।
सर सारे डाउनलोड करेंगे क्या?
सब यहाँ पे बस क्लिक है, जो भी फाइल हमें चाहिए यहाँ पे हमें जाके उसको सेव करना है, वो लास्ट में जाके एक साथ सारा डाउनलोड हो जाएगा। नेक्स्ट वी विल क्या PNG में जिसमें मन हो उसमें कर सकते हैं हम। जनरली हम प्रेफर करते हैं टिप में करें। Then we will do principal component analysis. Principal component analysis measure the variance in the sample and based on the variance in the samples, segregation between the groups will be done, Here you can see there is no clear-cut segregation between the disease condition and MOH condition, which is control condition and disease condition. So why this happen? This may be due to various factors. First one, if there is not significant change, there is less variance between the control and MOH group, then you will not get clear segregation between the both groups. Other reasons may also be like if you have within the variance within the group, then in that case you will also not get the clear cut segregation between the groups. So based on PLSDA analysis, PCA analysis, our control and MOH group are not clearly segregating. It means there is not enough variance between those two groups, and that is why in PCA analysis, control and disease condition is not segregating. So in option to that, we can also use PLSDA analysis, partial least square discrimination analysis, which is supervised, and this PCA is unsupervised. In supervised, group levels is used. You have seen in the input file like control group and MOH group. So that group will be utilized in PLSDA analysis, and in PCA analysis that group level will not be used. PCA analysis is generally used for exploratory analysis, where you don't care about the group name. This is the score, 2D score plot of PLSDA analysis. Here you can see clear cut segregation between the control and MOH group. This signified that there is, based on PLSDA analysis, there is clear cut segregation between control and disease condition. It means there are certain proteins which are overexpressed or downexpressed in case of MOH, and that expression of protein segregating this from control to MOH. Here you can also change the color of the graph. This is control, select, and from here you can select whichever color you want. And submit, and again you update, then you will get the required changes, and then save this image. In PLSDA analysis, proteins or features which are contributing to difference between the control and disease condition, and how much it is contributing, based on that PLSDA analysis give a score to the protein, and so and that score is called variable importance projection score. So based on this VIP scores, these are the important proteins which are responsible for segregating this control and disease condition. So this first upper one is this VIP score is 10. It means if you want to further do study on these proteins, so on basis of which you will select, you can select based on the basis of VIP scores. Means it is altering more in case of disease condition, these proteins. Then you can also draw dendrogram. Here you can see MOH and control samples are mixed, and this is the reason we are not getting clear-cut segregation between control and disease condition when we are analyzing our data using PCA. So if you have more number of samples, I have just 6 samples per group, and if you have more number of samples, so you can select the important HES3 from the data. And in control case, if suppose 2 or 10 MOH samples are coming towards the control, then you can remove that sample. Per group, you can proceed further analysis, and then you will get clear separation between control or disease control. Maybe disease development. We can also generate heat map. And these are the methods which are which methodology uses for creating this heat map. Distance measured by Euclidean and clustering method or the, and you can also set here you can, using this icon, you can generate the report. There is some error, report is not generating, so we will just download. These are the files related to the result of metabo analysis. Here you can see a folder, Excel sheet named volcano. Here you can access the differentially expressed protein. Here this is the accession ID of the differentially expressed protein, and this is raw fold chain and this is log fold chains, and this is arranged according to the raw p value, raw or log p value. So according to this protein is more significantly differentially expressed, and it depends how you will be using. If you consider fold chain, then you can arrange like this. Select this fold chain and sort it from largest to smallest. And then you can see the proteins which has highest fold chain in case of disease condition. So you can use fold chain, or you can log or you can p value. 0 स्टार 3 स्टार 2 स्टार मालूम है p वैल्यू में? अगर दो सैंपल ग्रुप है हमारे पास, उसका जो p वैल्यू है अगर 0.05 है तो उसको हम 1 स्टार देते हैं, ठीक है, और लेस देन 0.05 है तो उसको हम 2 स्टार देते हैं, और कहाँ तक 0.01 तक उसको हम 2 स्टार देंगे। अगर 0.01 से भी अगर कम है तो उसको हम 3 स्टार देंगे। तो ये जितना ही ज़्यादा सिग्निफिकेंट चेंज होगा ये p वैल्यू उतना ही कम होगा, ठीक है। तो हम इसके बेसिस पे भी सिलेक्ट कर सकते हैं जो p वैल्यू है। तो इसमें हमें क्या सिलेक्ट करना होगा? अगर हमें सबसे सिग्निफिकेंट चेंज देखना है तो सबसे लोअर वाला कहाँ होना चाहिए? ऊपर होना चाहिए। सो दिस प्रोटीन, इसमें जो p वैल्यू है इस प्रोटीन का सबसे ज़्यादा है, और सिमिलरली धीरे धीरे कम होते जाएगा। अब हमें ये फाइंड आउट करना है कि which are these proteins. For that, select this entire protein list. And open UniProt. In UniProt, we will do ID mapping, and in this section, just paste all those accession IDs and hit map IDs. रिपीट हो जाएगा। जिप के बाद से कोई भी जिप फोल्डर है उसको हमें एक्सेस करना है तो पहले हमें उसे एक्सट्रैक्ट करना होगा। एक्सट्रैक्ट करने के बाद से तो हम उस सारे फाइल को एक्सेस कर पाएंगे। एक्सट्रैक्ट कैसे करना नहीं मालूम? बता। क्या? यही ना, इसमें सॉर्टिंग हम फोल्ड चेन के बेसिस पे कर सकते हैं। किसे हमें ऊपर रखना है वो यहाँ से हम सिलेक्ट करेंगे जिसके बेसिस पे हमें करना है, और यहाँ पे सॉर्ट एंड फिल्टर का ऑप्शन है, वहाँ पे हम क्लिक करेंगे। सॉर्ट स्मॉलेस्ट टू लार्जेस्ट और सॉर्ट लार्जेस्ट टू स्मॉलेस्ट। तो अपने अकॉर्डिंग हमें जैसे एनालिसिस करना है हम सेलेक्ट करेंगे एंड देन सॉर्ट। वैल्यू को हम सॉर्ट करेंगे ना तो आईडी सपोज चेंज होगा हियर 801 देन इफ चेंज हुआ। ये वाला पार्ट मालूम है? यहाँ पे इसमें आईडी मैपिंग जो ऑप्शन है उस पर हम क्लिक करेंगे और उस बॉक्स में वहाँ से हम जो लिस्ट है प्रोटीन का उसको कॉपी करेंगे। ये उसके बाद से इसको हम यहाँ से एक्सेस कर पाएंगे। कॉपी पेस्ट के बाद से मैप आईडी नीचे सेक्शन में है राइट साइड में। हाँ, मैप 47 आईडीज। These are the list of proteins which are altered during disease condition, and using this download button you can download. You can also download in FASTA format, but for our analysis this FASTA format is not required. We will use Excel's format. Completed then back में बता। पेस्ट के यहाँ से नेक्स्ट मैप फॉर IDs. From here you can access those files. This is the link for that. It will take some time to complete. When if this completed, then you can paste these links. And then download this all this protein list by using download name, and then format would be Excel format, and we don't need to compress the data, so we select no.
ओपन करने के बाद क्या करना था?
लिंक ओपन करने के बाद से डाउनलोड सेक्शन है ऊपर, वहाँ से डाउनलोड करना है।
अच्छा, डाउनलोड।
हाँ, डाउनलोड सेक्शन पे जाएंगे और उसके बाद से फॉर्मेट में हम सेलेक्ट करेंगे एक्सेल। फास्टा फॉर्मेट में हमें नहीं करना है, और कंप्रेस की हमें कंप्रेस करने की ज़रूरत है नहीं, सिंगल फाइल है, ज़्यादा साइज है नहीं, तो कंप्रेस करने की ज़रूरत नहीं है, और डाउनलोड करेंगे।
compress field magic.
Here you can see the protein names and also gene name. We can copy this protein name, all protein name, and copy and then paste in the list of volcano plot. हमें यहाँ पे पेस्ट करते समय बस हमें ये ध्यान देना है जिस सीक्वेंस में हम यहाँ से कॉपी किए हैं हमें वैसे ही रखना है। अगर हम सॉर्ट किए हैं फोल्ड चेन के बेसिस पे लार्जेस्ट टू लोवेस्ट, तो उसको हमें वैसे ही रखना है हमें, और उस लिस्ट को कॉपी करके और यूनिप्रोट से हमें उसका नाम आइडेंटिफाई करना है। और सेम हम आके यहाँ पे एक यहाँ पे हम ऐड कर सकते हैं कॉलम, यहाँ से insert entire column, and in this column you can paste. So protein name and its accession ID, हाँ वहाँ से हमें क्या करना है, हमें तो स्टडीज का करना है, तो हम क्या करेंगे सब फोल्ड चेन जो चेंज है वो और प्रोटीन का नाम हम साथ में लाएंगे। तो उसके लिए क्या करेंगे, हम वहाँ से प्रोटीन नेम को कॉपी किए और जो वॉल्केनो प्लॉट का जो हमारा एक्सेल सीट है वहाँ पे हम पेस्ट कर देंगे। ठीक है, पेस्ट करते समय हमें यही ध्यान देना हमेशा जिस कंडीशन में हम इसको यहाँ से कॉपी किए हैं, ठीक है, उसी कंडीशन में ही ला के हम यहाँ पे इसको पेस्ट करेंगे। ये नहीं कि हम जब हम वहाँ पे आईडी मैपिंग के लिए गए तो हम इसको सॉर्ट किए थे स्मॉलेस्ट टू लार्जेस्ट, और पेस्ट करने के समय हम लार्जेस्ट टू स्मॉलेस्ट करके पेस्ट कर रहे हैं, तब वो रिस्पेक्टिव जो एक्सेशन आईडी रहेगा ना वो गलत हो जाएगा। तो जिस प्रोटीन का एक्सेशन आईडी है हमें वहीं पे पेस्ट करना है ताकि हमें करेक्ट इन्फॉर्मेशन मिले। एनालिसिस पार्ट ख़त्म हो गया है। आप लोग अगर पूछना है तो पूछ सकते हैं। यहाँ पे जो हम मिसिंग वैल्यू इम्प्यूट करते हैं, मिसिंग वैल्यू इम्प्यूट जो करते हैं, यहाँ पे हम जो इम्प्यूटेशन मेथड है वो हम अक्रॉस द सैंपल करते हैं। हम जैसे जब मैं पेपर को मैं भेजा था तो वहाँ से कमेंट आया था कि जो भी आप मिसिंग वैल्यू इम्प्यूट कर रहे हैं आप उसको ग्रुप वाइज कीजिए, ग्रुप वाइज करके देखिए कैसा आ रहा है। तो ग्रुप वाइज करने में क्या इशू हो सकता है, आप लोग कुछ बता सकते हैं? ग्रुप वाइज group wise in both cases will be column wise. I mean group wise. You are doing a missing value imputation of group 1 separately and then for other groups. We are using for missing value threshold 30%. So इसमें इशू ये हो सकता है, मैं बता रहा हूँ, मैं हमारे पास 12 सैंपल है, ठीक है, तो एक पर ग्रुप 6 सैंपल है। तो अगर एक ग्रुप में हमारा 30% से ज़्यादा है, ठीक है, 30% मिसिंग वैल्यू, सपोज करिए एक ग्रुप में हमारा 6 सैंपल है, उसमें 30% से कम मिसिंग वैल्यू है, तो वो प्रोटीन हमारा वहाँ पे क्या रहेगा, डेटा में रहेगा, ठीक है। लेकिन अगर वही जो दूसरा ग्रुप है हमारा, वहाँ पे मिसिंग वैल्यू 30% से सपोज कीजिए ज़्यादा है, तो उस केस में क्या होगा, वो प्रोटीन वहाँ पे रहेगा नहीं, वो उस वहाँ से वो एलिमिनेट कर दिया जाएगा। तो फर्दर लास्ट में हम क्या इंटरप्रेट कर पाएंगे, मिस इंटरप्रिटेशन ये होगा हमारा कि ये सर्टेन प्रोटीन कंट्रोल केस में एक्सप्रेस कर रहा है लेकिन डिजीज कंडीशन में एक्सप्रेस नहीं कर रहा है, जो कि क्या है, फॉल्स है। ये मिसिंग वैल्यू आता क्यों है, मिसिंग वैल्यू आने का रीज़न है ये इंस्ट्रूमेंट का ये अपना ये लिमिटेशन है कि आप कैसे भी सैंपल प्रिपेयर कर लो, ये कुछ ना कुछ मिसिंग वैल्यू ये आते ही हैं, ठीक है। तो इस तरह से हम गलत इंटरप्रिटेशन हमारा हो सकता है, तो इसीलिए हम मिसिंग वैल्यू अक्रॉस ऑल द सैंपल हम करेंगे।
डेटा यूनिक प्रोटीन फिट करने के लिए लिया, हमने पर्टिकुलर वोल्केनो वाले एक से 2 लिया, कोई और
वोल्केनो प्लॉट से जो इसलिए हम लिए क्योंकि वही है क्या है जो सिग्निफिकेंटली डिफरेंशियली एक्सप्रेस प्रोटीन है, वो उसका लिस्ट है उसमें वोल्केनो में, ठीक है। वोल्केनो में हमें क्या मिला, 47 डिफरेंशियली एक्सप्रेस प्रोटीन, 6 डाउन रेगुलेटेड एंड 41 अप रेगुलेटेड एंड 1800 समथिंग आर नॉन सिग्निफिकेंटली अल्टर्ड। सो from that we are getting this 47 differentially expressed protein.
जो भी हम स्टैटिक्स देख रहे हैं उसमें सबसे ज़्यादा डिफरेंशियल चीज देख रहे हैं हम उस एक्सेल।
हाँ, बाकी जगह पे हम कुछ और एनालिसिस कर रहे हैं, जैसे पीसी हम कर रहे हैं किसके लिए, ग्रुप वेरिएंस चेक करने के लिए, ठीक है। पीएलएसडी हम क्या कर रहे हैं, क्लासीफाई करने के लिए कर रहे हैं हम यूज़, ठीक है। हीट मैप हम किस लिए यूज़ कर रहे हैं, हीट मैप का क्या यूज़ है, हीट मैप से हम क्या कर सकते हैं कि एक ही साथ सर्टेन प्रोटीन्स को लेके हम उसका हम एक्सप्लोर कर सकते हैं, क्या एक्सप्रेशन पैटर्न है दोनों ग्रुप में सैंपल वाइज, एक साथ हम चेक कर सकते हैं बाय गोइंग टू द डेटा, बाय जस्ट सिंपली सीइंग द कलर्स, ठीक है। तो इसलिए हम हीट मैप करते हैं। तो सारे से जो भी एक्सेल शीट आ रहा है हमें सबकी ज़रूरत नहीं है, हमें आगे अगर हमें फर्दर स्टडी करना है, अगर हमें क्या करना है, सॉर्ट आउट करना होगा प्रोटीन, इसको हम करके क्या करेंगे, इससे हमें 47 मिला, हम 47 में से भी फर्दर हम सॉर्ट करेंगे, और वो सॉर्ट करने का क्राइटेरिया बहुत सारा हो सकता है। एक हो सकता है कि हम फोल्ड चेन के बेसिस पे सिलेक्ट करें, तो हम क्या ले सकते हैं, जो हायर फोल्ड चेन के हैं उसको ले सकते हैं, या सबसे ज़्यादा जो लोअर है उसको हम ले सकते हैं। उसके अलावा क्राइटेरिया क्या हमारा हो सकता है कि जो हम पी वैल्यू हम ले रहे हैं, तो सबसे ज़्यादा जो सिग्निफिकेंट है, जो मैं बता रहा था 1 स्टार 2 स्टार 3 स्टार, तो उसके बेसिस पे हम ले सकते हैं। उसके अलावा क्राइटेरिया क्या हो सकता है कि जो हमारे पास से 47 डिफरेंशियली एक्सप्रेस प्रोटीन आया है, इसका हम यूज़ करें प्रोटीन नेटवर्क जनरेट करने के लिए। अब वहाँ से हम स्ट्रिंग डेटाबेस में गए, हम वहाँ से प्रोटीन प्रोटीन इंटरेक्शन हम चेक किए। अब उसके बाद से हम क्या करेंगे, उस जो भी इंटरेक्शन पैटर्न आया है उसको हम एक्सट्रापोलेट करेंगे किसमें, साइटोस्केप में। और साइटोस्केप में ऑप्शन होता है कि हम ये पता कर सकते हैं कि कौन सा प्रोटीन कितने अदर प्रोटीन से इंटरेक्ट कर रहा है। मैन्युअली हमें दिखेगा, लेकिन हम उसको काउंट करने में थोड़ा एक्यूरेट नहीं हो सकते हैं, तो उससे हम करेंगे तो एक्यूरेटली वो मेजर करेगा कि कौन सा प्रोटीन किससे ज़्यादा किस किस प्रोटीन से इंटरेक्ट कर रहा है। सपोज कीजिए कोई एक जो मतलब कि जो भी प्रोटीन सबसे ज़्यादा प्रोटीन से इंटरेक्ट करेगा तो हम उसको क्या बोल सकते हैं, क्योंकि ज़्यादा इंपॉर्टेंट प्रोटीन है। अगर कोई एक प्रोटीन है जो 1-2 से ही इंटरेक्ट कर रहा है तो उसको हम ज़्यादा इंपॉर्टेंट प्रोटीन नहीं बोल सकते हैं। तो इसके बेसिस पे भी हम सेलेक्ट कर सकते हैं। और कुछ बहुत बात तो हमें होता है ना, सब कुछ हमारे सामने दिखता है लेकिन ये नहीं समझ में आता हम इसका आगे लेके करेंगे क्या, हम क्या इससे समझ रहे हैं। तो अगर इस तरह का प्रॉब्लम हो तो बता सकते हैं आप लोग। मुझे स्टार्टिंग में नहीं समझ में आता था, इससे हम जानना क्या चाह रहे हैं। ये तो सब कुछ प्रोसेस है, किसी को भी बता देंगे वो 1 बाय 1 कर लेगा, लेकिन उससे हम क्या इन्फ्रेंस निकाल रहे हैं सबसे ज़्यादा इंपॉर्टेंट है हमारे लिए। हमारे लिए कैसे हो रहा है वो ज़्यादा इंपॉर्टेंट नहीं है, वो कोई दूसरा भी कर सकते हैं। हमारे लिए अगर जो बायोलॉजिस्ट हैं जो स्टडी कर रहे हैं उनके लिए सबसे ज़्यादा इंपॉर्टेंस है कि इससे हम क्या इन्फ्रेंस निकाल रहे हैं। तो वो अगर नहीं समझ में आ रहा तो आप लोग पूछ सकते हैं। नहीं, इस जो भी होगा ना इसी के आसपास ऐसे ही आएगा ना। क्या कैसे रिलेवेंट है, कैसे रिलेवेंट है, इसको तो करने के लिए हमें स्टडी करना पड़ता है। इससे हम ये आइडेंटिफाई कर रहे हैं कि कौन सा प्रोटीन कितना ज़्यादा अल्टर हो रहा है उसके एक्सप्रेशन पैटर्न में। अब कैसे वो क्या उसका रोल है उस डिजीज कंडीशन में, वो आपको स्टडी करना पड़ेगा। और उसके अलावा आप अगर आपके पास लिमिटेशन है, आप सारे प्रोटीन के साथ एनालिसिस तो कर नहीं पाएंगे। हमारे पास 47 प्रोटीन है, ठीक है। और सपोज कीजिए हमारा इसमें सबसे ज़्यादा जो फोल्ड चेंज है होमर प्रोटीन होमोलॉग 2 का है, ठीक है। और हमें इसका फंक्शनल कैरेक्टराइजेशन करना है, ठीक है। और फंक्शनल कैरेक्टराइजेशन करना है लेकिन हमारे पास इसके लिए एंटीबॉडीज़ अवेलेबल नहीं है, ठीक है, तो हम इसको नहीं कर पाएंगे। तो उसके लिए ये सब बहुत सारे पैरामीटर्स हैं। हम एटलीस्ट 10 प्रोटीन को हमें सेलेक्ट करके रखना होगा जिसको हम आगे जाके हम स्टडी कर सकें। और उसमें से किसके लिए हमारे पास रिसोर्सेस अवेलेबल हैं हम उसको कर सकते हैं। अगर डाउन रेगुलेटेड प्रोटीन का आपको फंक्शनल कैरेक्टराइजेशन करना है, उसका क्या रोल है, इसको अगर करना है तो आपको क्या करना होगा, एक दूसरा जब ग्रुप आप बनाएंगे तो उसमें क्या करना होगा, उसमें आपको इसके एक्सप्रेशन को ओवर एक्सप्रेस करना होगा। और ओवर एक्सप्रेस करने पे क्या चेंजेस आ रहे हैं उसको स्टडी करना होगा। हमारे केस में क्या है, हमारा ब्रेन पे हिपोकैम्पल प्रोटियोमिक्स किए हैं, तो हिपोकैम्पस पे क्या क्या चेंजेस हैं हमें स्टडी करना है। तो मैं क्या किया हूँ कि मैं इसमें से एक फोकल एडिशन काइनेज 1 प्रोटीन है उसको मैं सिलेक्ट किया हूँ जो कि अप रेगुलेटेड प्रोटीन है। और उसको मैं क्या किया, उसको डाउन रेगुलेट किया यूजिंग इनहिबिटर Y15। और उसके बाद से मैं चेक किया कि इनहिबिट करने के बाद से जो न्यूरोनल अर्बोराइजेशन है, क्योंकि जितने ही न्यूरोनल अर्बोराइजेशन रहेंगे अच्छे से उतना ही जो कम्युनिकेशन है न्यूरॉन्स के बीच में ईजिली होगा, आसानी से और अच्छे से होगा। लेकिन न्यूरोनल कनेक्शन अगर ब्रेक हो जाते हैं, डैमेज हो जाते हैं, तो उस केस में न्यूरोनल कम्युनिकेशन प्रॉपर नहीं होगा और ब्रेन प्रॉपर फंक्शन नहीं करेगा। तो उस केस में हम क्या किए हैं, उसकी एक्टिविटी को डाउन रेगुलेट किए हैं, इनहिबिट किए हैं यूजिंग अ केमिकल। और आप अगर डाउन रेगुलेटेड का अगर लेके कर रहे हैं तो डाउन रेगुलेट में क्या करना होगा, अप रेगुलेट करना पड़ेगा। तो आपको पहले ये देखना होगा कि आपके लैब में वो उस तरह का स्टडी करने का स्कोप है कि नहीं है। अगर है, आपके पास लेंटिवायरल वेक्टर सिस्टम है, वो सब आपके लैब में है, उसको कर सकते हैं, एक्सप्रेस कर सकते हैं, तब उस वाले भी प्रोटीन को आप सेलेक्ट करके उसका फर्दर स्टडी कर सकते हैं। अगर नहीं है तो आपके पास आपको सेलेक्ट करना होगा कि किस पे हम कर सकते हैं। तो ये सारे भी पैरामीटर्स हैं जिसको ध्यान में रखते हुए हम प्रोटीन को हम सेलेक्ट करते हैं। हो गया तो हो गया, बस खत्म ही है। भाई लोग जैसे
क्वेरी पूछ लीजिए, 10 मिनट बाद हम टेस्ट कर देते हैं।
ठीक है, ठीक है,
45 मिनट।
ठीक है, ठीक हो गया, ठीक है।
So everything I've shown you is about learning or looking for, using machine learning to look for patterns in data. Sorry. So now we're going to go on to Weka itself. Obviously, just for those who are watching and are not in the room, I've just been talking around the package manager for Weka, referring to deep learning packages.
So it would be much better if you could just put it on the slide.
Okay, sure. Okay, is that better?
Yeah, thank you.
Okay. So, I've just been talking about deep learning for J. I've just been talking about AutoWeka as being useful tools to install. So there are various packages or applications within Weka, and they implement machine learning in different ways. So the first one, and the most easy to use and the most commonly used one, is Weka Explorer. And this is a GUI, so it's a graphic user interface. It means you can put things in place, implement solutions, and you don't need to do any coding. So what I'm going to do, first of all, I need to load a data set in. Some important things to know. I'm just going to go to my PC, and I just want to, right.
Okay. So just switch it off. That's why the sound is on.
Shall I try one of these?
Maybe you can try. Just turn on.
And just something particular to Weka that you have to do. Oh, this is awful. Maybe don't need the volume down or something. Is that better? The key thing to note here is where we have a class in Weka, we have to use a word to denote the class rather than a number. So normally we would use binary notation. 0 is one class, 1 is another. Here we use a word. So pos means disease positive, neg means disease negative. That's the only real difference. Everything else in Weka can be loaded as a CSV in the same way. So I need to load some data. So first thing I do is open a file. Just go up to the relevant directory. Right. Data for the default file type for Weka is an ARF file, but I'm using CSVs because they're more transportable. So I'm just going to load data for course Weka. So you can see the data set, and what it's doing is giving distributions of various variables and the number of individuals who are in a negative, disease negative and a disease positive class. So in this case, red is negative, blue is positive. So essentially it's providing me histograms just so I can see what my data looks like. So that's the file load interface. I can choose filters for that. So I can use smoothing filters. Say we're using mass spec raw signal, you could smooth those signals out here with filters. So I can load those filters in. I'm not going to use them now, but there's lots of ways of filtering data in Weka. There's not time to go into those right now. So what I'm also going to do, first off, is I'm going to select all the variables, and they're the ones I want to use. So by selecting those tick boxes, it means I'm going to use these for the analysis. Right, so now I move from the pre-processing tab up the top here to a classify tab. Now, again, I can choose one of many classifiers. You see here I have Bayes classifiers, I have functions, so I've got a multi-layer perceptron here, I've got deep learning as a classifier if I want to use deep learning, simple logistic regression, many other things. I can also use rule-based clustering, 0R and part of those, many other types as well. And you can see here I've got decision stump, I've got random forest, I've got random tree, I've got J48, which is another classifier based in Java. Nice thing is, every time I move over one of these, it tells me what it is and how the data should be formatted for that. So I'm going to start off, I'm going to make a random forest. So now I've got this random forest. If you look here, this is the parameters, the hyperparameters for that random forest. I'll stop it. So if I click on this, I can immediately start changing the hyperparameters in this. If I look at the help for Weka, it's not immediately here, but if you look it up in the help, say random forest, it will tell you what these mean. But if you move the cursor over, it does give you an indication as well. So for example, number of iterations, how many trees am I going to make in the forest? If I look at number of decimal places, that's just rounding. If I look at the batch size, this is the number of instances to process. I can print the classifiers, I can do various other things. I can also save this if I want to. So if I've got a particular form of hyperparameters that work, I can save them here, or I can open new ones. Well, I'm just going to click OK. What I also have here is the testing options. So I can use a cross-validation, which is like a bootstrapping. I can use 10-fold cross-validation, so 10 different splits of the data. I can also use a percentage split. So if I want to train on 66% of the data and use 33% as testing data, that's where I can do it. I need to set, I can have other options, there's a whole load of other stuff I can change here. I need to set the class that I'm predicting. So I'm basically selecting the output class here, and I've set it all up. So what I now do is I click start, and you can see it's quite quick. It's a small data set, but I've got immediately various things. So I've got how the classifier has been set up. I have the number of iterations, things like that. Then I have the correctly and the incorrect. What? Dear me. What is going on with this machine? So I've got the correct and incorrect classes. I've got the error, and I've got the true positive rate, false positive rate, the rock area. So I can immediately see how well my model is doing with that method. So I can try that out. The other thing I can do is, if I right click on this box here, I can save this model. I can say load a model. I can visualize the errors. I've got a cost curve. I've got rock curves. I can do a lot of other things with the result from that. So it's quite a simple interface to use once you've seen how to do it. So that's the most basic form. What I can also do in here is if I click on select attributes, what select attributes does is it allows me to iteratively work through the data and find the best subset of inputs. It's this parameterization that we can use. So what I'm going to do, I'm going to choose the attribute evaluator, and I'm going to check the wrapper subset evaluation. So a wrapper method, it basically uses the performance of an algorithm to select parameters. So we have two forms. Effectively we have a filter method which looks at the performance of individual features and selects the best ones from that. And then we have a wrapper method which uses an algorithm performance. What we're essentially using here is a combination of a filter and a wrapper. So we use the algorithm performance in the filter to select the best parameters. So what it does is we can use, for example, if I click choose here, we can use a random forest as the basis for our parameter selection. And then we can choose the search method. So this is various search methods. Best first looks at simple consecutive non-improving nodes, as it says. We've got a greedy stepwise, and that's a bit more aggressive in what it selects. And then we have a simple ranking tool as well. Yes. So I set, as I did before, my class. I can put cross-validation into this as well. So I can use cross-validation to check how it's doing over multiple views of the data. And I'm not going to do that because it takes more time. But if I click start, it's run through it already. So for a random forest, it's put 6 attributes forward, and then okay, did it work? It didn't work for that, so I'm just going to choose a different one that I know works, which is a multi-layer perceptron. Click OK, click Start. So when we know it's running, we can see the Weka moving at the bottom there. It's put 6 inputs forward. It's predicting disease versus healthy. So it's taking a little bit of time because it's doing thousands and thousands of calculations. And if you can see, it's now identified the 4 best attributes as TNF-alpha, IgG, ER, MMP9. So it's searched through all those combinations, and those are the 4 best ones that it's found. So if you've got machine learning data, if you've got proteomics data, you might want to filter it a bit first, but if you wanted to select, say, from 200 or 300 features to find the best combination, you could use that on that data to see which is the best combination of things that predicts a answer to a question. Using whatever algorithm you need to use. It's not having a good time today. So I could then go in and rerun this algorithm with just TNF-alpha, IgG, ER and MMP9, and it would hopefully improve the performance. So we can use this as a parameterization tool. We've got a huge amount of flexibility in the algorithms we use. We've got a fair degree of flexibility in the search method we use as well. So that is one way of analysing our data. I'm going to show you some more that you can use with Weka. The next one I'll just show you is Auto Weka. So here what we do is we're literally setting up an experiment and telling it what to look for. So this tells us how many things to look through, batch size, how much memory to use, the number of best configurations. So this is just looking for the top single best configuration out of them all. But maybe we don't want to just rely on one. We might want to select 5 or 10. Give us the top 10 rather than just the top 1. The seed is a random number seed. So basically, if we run this with the same random number seed for the same data for the same time, we probably get a very similar or the same answer. The random number seed is how it's randomized, that we can fix the way it's randomized by using that particular seed. Change the seed, you'll get a different set of results. Because it's a randomized process, the way the cases are selected, the way the weights are initialized, there's a lot of randomness in there about setting up. But that can be an advantage. So if we find things that are there despite that randomness, then they're more powerful. Because ultimately it's starting off with a random state and then finding the correct state. And if we keep finding that same correct state, then it's going to be reinforced. So I'm going to not run this for 15 minutes. I'll run it for 5. And if I click OK, I want to use the same output. And then I just click Start. And as with all best machine learning algorithms, not much happens on the screen because you don't want it to, because it's a waste of resources. But working in the background, that's telling me it is. This is now looking at a whole load of different algorithms, different solutions. So this is a good time to actually run it while they're fixing the microphone. So this will just keep running. It tries lots of algorithms, lots of parameters, and it will find the best solution for this data set. And sometimes that's all you need to do. You've got a data set, you want to find the best method to use, and maybe then you can use that as a basis for further data sets that you evaluate. So that's helpful. I can't really show you how to do it because I'm doing it on a machine now, I don't want to change it, but you can tweak the settings of Java, it's not done in Weka, to increase the memory limit of Weka as well. I think it currently uses 16 gigabytes of memory, but you can change that to, if you've got a really powerful machine, 64 gigabytes of memory. That gives you the potential to load pretty large data sets into memory and let Weka run through. Now you can also tweak Java and Weka. I don't know how to do this because I've not needed to, but you can tweak it to run on a GPU. Okay, thank you. So if you tweak it to run on a GPU, you're going to get a speed up of performance because it's parallelizing it as well. So where have we got to? We've run 98 different models so far, and we've got about 23% error is the current error rate. That's pretty bad actually. Well, it is a pretty rubbish data set. I made it to this purpose and wanted it to be noisy. So it's running, we're finding results. We can come back in a little bit and see what we're getting. Right, while we're doing that, I just want to show you another way of doing this. So in this folder are files which are part of a bigger file. We recently had a paper published on muscle aging, and this is data from that muscle aging study. But what I've done is the data structure would normally be, we have the genes across the top and the cases down the side. And we might have 20,000 genes along the top and 100 cases, 200 cases down the side. Because Weka can't really cope with that in its current form, what I've done is I've basically taken the column header, I've taken the gene expression value through the population for a particular gene, and then I've taken the state to whether it's young or old. And well, in fact, I think for this I use regression. So it's the absolute age of the person where the muscle biopsy was taken. So by splitting the data into one file per gene, that means it's more efficient. It's not loading the whole data set into memory, it's just running one data set at a time. So if you have any command of R, it's actually quite easy to split that up. You could, if you have plenty of time on your hands, you could split it up manually, but by getting R to literally extract the columns into files, you could probably do that with 3 lines of code. You could probably get ChatGPT even to write you the code to do that. But by splitting it up, you can use Weka as a way of looking through transcriptomic or proteomic data for enrichment. You want frequently the first time to enrich for a set of genes that stably answer your question. So if something predicts something, then that's interesting. So if this gene predicts muscle age, that's interesting. But in the genome, in all likelihood, there are multiple things that are going to be associated with muscle aging. And what I want to do is find from across the whole genome those things that are associated with muscle aging in a stable way. So if I look at a different randomization of the data, I will find the same gene in the top 100, 200, 300 genes. If I repeat it again, by looking at the data and coming back to the same set of genes, I will, if the same things keep coming out, I know they're important. And I know they're unlikely to be false discoveries. So this particular workflow is centered around that. And once the other part of Weka is finished, I'll show you how to use it to enrich. So, what these individual files look like is this. So it's literally a column called case, that's the individual in the population. Within that we can randomize as well, and we will. We've got the expression, and the header of that column is the gene name. And this is using GIAN notation. And then we've got the column header for age of the person who gave the muscle sample. And they range between individuals who are 20 and individuals who are, the oldest one I think is 84. So this is what we were doing in this study, understanding the impact of age on muscle, and particularly identifying risk factors for sarcopenia, which is what muscle wastage in old age. So I can use this data, and you see what I've done here is each one of these in this folder is a separate gene, and I can run through these and compare how they perform in an analysis workflow. Okay, any questions on what I've shown you so far? Hopefully it's being videoed, hopefully you'll be able to hear me on the video. Make notes while you're going on, take pictures, take videos if you want to for your own use, I don't mind. So, our Weka Explorer is finished. It's run for 5 minutes. The rock area for this combination, so what did it find? Well, let's have a look. It found that in this case the best classifier was a classifier called a rule-based classifier called a JRIP. It's got the parameters for that classifier here. The error rate here, and then I can actually, if I wanted to instantiate that algorithm with those parameters into Java, I can use that string of code there. So that will set that up, as long as the right libraries are in place, in Java, and run that same algorithm. So if I could put it into an algorithm for somebody else to use. So that's that bit. It gives me the usual metrics, classifier rate, it gives me correct cases, 215 correct, 47 incorrect. So that is an improvement on what we had before. It also has a confusion matrix. And let me explain a little bit about confusion matrices. They use the classifiers and they tell me how many are right and how many are wrong. But it's a bit more than that. So we know A is positive, B is negative. Let's move the cursor out of the way. So of the positive cases, 35 were predicted as A and 37 were predicted as B. So that immediately tells me my algorithm is not so good at predicting positive cases. Yeah, it's got more wrong than right for the positive cases. If I look at the negative cases, it predicted 180 as B, and it predicted 10 as A. So it only got 10 of the negatives wrong. So that immediately suggests there is a bias to my results. And I deliberately put that bias in because I wanted to talk to you about balance and bias in classifiers. So for example, if you look at the number of cases, well, I've got 190 negatives and 72 positives. That means the negatives are much more represented in my data set than the positives, which means that the algorithm is going to encounter negatives a lot more of the time. So what I have to do is when I build a classifier, I have to balance the numbers, ideally. I can probably get away with 2 times versus 1 time, say 2 times A, 1 times B. But much more than that, I start to introduce bias. And I've had people who come to me to analyze data where they've got 99 of group 1 and 1 of group 2, and I just say, you're not going to get anywhere with that because you need a good representation across both of the classes for the algorithm to work well. So that's an important consideration, balancing the numbers. And this is not a balanced study. If you're working on continuous values, you don't have to worry about balancing them because you have a continuous range. That's okay. So that's a it just says if I want to get a better performance, I run it for longer. Well, I kind of knew that. But I would say to get a better performance, I should balance the numbers and run it for longer. Any questions about any of that? Okay. So the other thing I can do, I just want to show this. This is specifically for image analysis using deep learning. So what I can do here is I can, if I label images with a class name in a folder. So let's say I have, well, one of the most weird ones I've seen is people who are trying to distinguish between a fajita and a chihuahua. So they've got these fajitas, which is like a rolled flatbread rolled up with cheese in Mexican food, and they have a chihuahua, which is a small dog. And what they did is if we were to run that, then we'd have a series of images which were called fajita 1, 2, 3, 4, 5, 6, 7, 8, 9, and we'd have a series of images called chihuahua 1, 2, 3, 4, 5, 6, 7, 8, 9. And we would put them into a directory, and then we could, where it says open image, we could utilize that to pull in those images, and it would automatically convert them into arrays of pixels. So it converts them into something the algorithm can use. What I can also do is I can use the GPU for this. I just have to do a bit of Java setup. It tells you how to do that. I haven't actually set that up on my machine because it messes with another program I use. It will show, once you've loaded images, it will show them in here. And then I can run, so I've got various algorithms, but the only one I can run in this is deep learning for J, and it's a convolutional neural network, a CNN. So this is previously set up a default convolutional neural network in here for me. I basically have pre-trained zoo models, which it's set up some weight settings that are good for image analysis. It has I've got a model output decoder, so what that will do is it'll give me the convolutional layer components and predict something. And then I can just run it. So immediately you've got access to CNNs for analysis of images, allowing you to utilize them without, they normally would code these in Python a while. So that's a useful tool. Say you're looking at cell images, say you're looking even at imaging mass spectrometry, potentially you could use CNNs on that. So that's another technique which is available in Weka, and I'm not going to run it. I'm not going to run it right now, so I seem to be getting a lot of emails through today. So okay, that's the most simple interface for Weka. That's Explorer. I recommend you have a look at the videos, that you have a look at how to use it. You have a lot of help, a lot of support. If you want to try something, you can just Google, how do I do this in Weka? And I'm sure there's probably a video for that, or some text for that. So I'm now going to get back onto looking at that data set that I mentioned to you. So instead of using Explorer, we use for that Experimenter. So Explorer is very much about you learning to use the algorithm, trying things out in a small scale. Experimenter is about running multiple algorithms for multiple data sets. So you can run Experimenter on 20 different data sets with 20 different algorithms and find the best results. It's a way of automating the whole process. It will allow you to do things like trying out 2 different algorithms on, let's say, like the files I have, 20 different files of mass spec mass spec values, answering a particular question. It will allow you to evaluate those algorithms on each one of those features, and it will give you the results in an Excel spreadsheet at the end of it. So you can kick out the results as a report, which is in a spreadsheet. Let me show you how to do that. So in the setup phase, I can open a previously set up experiment. I'm not going to do that now. I'm going to create a new one. So the first thing I'm going to do is put the results into a file. I'm going to call that file GIAN results, and I'm going to set that to go to my computer. Right, we have a GIAN course folder here. Oops. So I've set up where the results need to go. I'm going to use a training test split. My problem is a regression one because age is a continuous value. I'm going to just do 2 repeats, otherwise we'll be here for a very long time. And I'm going to set data sets first. So basically it runs the same algorithm on a particular data set again and again and again. Now when it comes to adding data sets, what I can do is go to my folder where my results are, and then I know they're all CSV files in here. No, they're not zipped files. And basically click the top file, press Shift to click the bottom file, and click Open. So it's now loaded all of those files into the experiment, and we'll look sequentially through those. The file name is the gene name. That's important because we need to, that's what's going to be written to the results file, so we can deconvolute that by looking at the gene name. And now what I need to do is select an algorithm to run on those. So I'm going to, for this I'm going to use an MLP. In the MLP, I can set the architecture up, and I can set it to automatically select the number of hidden nodes in the architecture. And I do that by just putting an A in here. So it'll automate that process. You can see also I can change my learning rate, my momentum. Not sure if I can change the transfer function. Click on false, will that round? I think it's not letting me change the transfer function because it's on auto build. If I want to, I can. So I'm going to use an MLP, and that's all set up and ready to run. I can add notes into my experiment if I want to. So I'm just going to click on Run now and click Start. So it's now working through those data sets, each gene being a data set, evaluating those in terms of their ability to predict muscle age. And we can use the correlation coefficient and the root mean squared error as the selection parameter for that. So it's working through, it'll take a little while. So, are there any things you would like me to clarify on what you've seen so far? Have a go, have a play. You know, there's data in Weka for you to have a go with. Use that. You will learn how to do it. A lot of people use Weka and it will produce publication quality results because of the power of the algorithms that are embedded into it. So it is useful. Yeah, it's just running through now. Takes a little time. A lot of my role as a machine learning specialist or computational biologist involves me waiting for bars to move across screens and folders to fill up with files. It's when the algorithms are running, you don't see much, but the nice thing is you can automate something and let it run for a few days and then come back and harvest the results, which is quite rewarding. Okay, it's finished. So good, that wasn't too slow. So now, if I go to the GIAN course, and here you can see we have a CSV file with our results in it. I'm not going to convert those for now. Well, you can see here, in this file I've got the key data set, so that's the gene name. I've got the algorithm. I've got the options set for that algorithm, version. I've got the date and time, which I think is a non-date format here. Number of training instances is 43 individuals. Number of testing instance 22. Now you see I've got my mean absolute error, root mean squared error, relative absolute error. What I'm really interested in is the correlation coefficient, because I want to find the things that correlate best with muscle age. So what I can do, the most simplest form is I can sort by correlation coefficient and sort largest to smallest. Click OK. So let's put a split in this. So across the different randomizations, the best gene in its own right is GI 10092672. You can see I have now, this one has then 1004, then 1019. Then I've got 1009 again, then I've got 1019, then I've got 1019, then I've got 1004, then I've got 1004. So you can see across the different instances, the same 3 genes are coming up as highly predictive of muscle age. So I put 50 in, and I can immediately see which ones are stably coming up. And in that way, by doing that, I can say, well, I can start to enrich and say these are the genes out of the 50 that consistently answer the question of muscle age. Now you can use that for mass spec data if it's in a protein name format. You can use that for expression array data. You can use that for RNA-seq data. It's a way of generating new hypotheses based upon particular data sets and particular questions. So, any other questions? That's all I've got to cover for Weka. There's a lot more to go at. There's a lot more things you can do. But that really kind of introduces the basics and how you can utilize it for the work that you potentially can do. Right, I'm going to end my full screen. Minimize that. Machine learning with a no-code solution in Microsoft Azure. Essentially, it's very similar to what I showed you in Orange. It's a workbench type solution. And then there is another piece of software called RapidMiner. And RapidMiner is an automated way of, a bit like Weka, of looking at with a no-code solution, looking at large data sets and pulling out features. So Rapid Miner, I think the free option only has a very small number of algorithms and arguments. You can pay a nominal fee and get a lot more. But I've shown Weka and Orange because unlike Azure and Rapid Miner, they're free. What else is out there related to this? Well, I mean, there's not time here to go through coding, but the two recommended systems for machine learning, I would say the primary ones are Python. So you can see there's lots of online courses on how to code machine learning in Python. The key I find with Python is you have to install and manage the environment. And for biology, I would do that with Anaconda. So you load Anaconda. Anaconda helps you set up an environment, loads all the necessary packages, and then you can install Python and it will have the necessary environment for what you want to do. You can also use R. So R is, I would say, much more biology friendly because you've got elements like bioconductor in there. Although machine learning wise, the library, which you've got PyTorch in Python and you've got Keras TensorFlow in Python. But with R, Keras is a later implementation. It's not so well developed. But you can still put machine learning algorithms in Keras. The way these things work, just to explain, you've got these libraries, you have to install and then load the libraries, and that gives you functionality that you can put into the code. So in order to do something, you load a library, and Keras is a machine learning library, so it allows you to write code that implements machine learning. There's also Tidyverse. Tidyverse is a massive library. It allows you to use functions that are very good at talking to one another. It's good at things like data manipulation, graph plotting, things like that. And then the last one is dplyr. And dplyr is very good for manipulating data arrays and spreadsheets and the like. But you know that's and the story behind that is quite long. So have a go, but the story is quite long. You can do things in Java. I'm not so familiar with Java, but it's less entry level. But in many ways, the libraries in Java, I think, are more powerful. They're also, and this is important for what we're going to consider soon, there are some very good network biology tools. Biology exists in a network. So one of the good tools is GeneMania. With GeneMania, you can just drop a list of genes or protein names, but if you use the gene symbol into that, set a species, and it will identify what interactions there are in those in the literature. So it's a way of looking at gene associations across networks. Yeah.
So Graham is showing you some tools, right? And if I am right, Shambhu would have shown you a list of differentially expressed proteome, right, Shambhu? Metascape, metabolist. So your job for the day is take a tool of your choice and read and use any of the tools that you find, especially network biology tools or Orange. You interpret the same data with your point of view. You see, the good thing about multi-omics data is even if the data is lying around for 5 years and you give it to 100 people, 100 people will use their own point of view, their own biology and expertise to get new insights from it. So your assignment, which will of course be judged, and this is for tomorrow morning before you reach, you can drop an email. You have the email, GIAN Proteomics and AI, And even the PDF that many of you are sending, please don't send me on the WhatsApp group. I'm getting spam. You just say assignment for day 3, so then we can skip the test part. Right? So
That's good.
it'll be good for
Yeah.
you to use this tool.
So you could use your differentially expressed genes, the values for those.
Yeah, on the mail, we just make a small PowerPoint presentation of maybe 5 slides. That this is what the differentiation of gene. You can ask Shambhu regarding what was the biology. You might have told me what this experiment was all about. You can go back to the original paper, Shambhu. Which paper was this? Experimental brain research. So you can go back to that paper. It's available. You can ask them to share the paper with you. And they will try to interpret something new, something novel that you find with your analysis.
Yeah, so you could drop those gene names or protein. The protein names as a SwissProt name won't work, but if you get from SwissProt the gene symbol, which is the short gene name, you can drop those into any of these, find the network biology. You could, if you've got the raw data, drop them into Weka and build a classifier that separates one group from another. Yeah. So if you've got that raw data, you could build a classifier in Weka using AutoWeka, or you could use parameter selection and find what was the best subset of those DEGs in combination that predicted the class. So you can see immediately there's a lot of ways of using this to explore your data further.
Maybe on the final day we can have a presentation randomly from maybe 5 people. We will see how the same data, 5 different eyes, look at it in 5 different points of view.
Yeah, I mean we could take the top 5 people from the quiz if they really want to. Then another one, and this is very much protein focused. So string is about protein-protein interactions. That's important for biology and for drug discovery. But if you're looking at proteins, you want to see how they interact in a network, and it's all about differential binding. It's about energetics and so on and so forth. This is still the one I just demonstrated Weka and Orange, so
Okay.
but I shouldn't be long. Maybe 10 minutes, 5-10 minutes. Yeah, okay. Yeah. So Network Analyst does something similar. Cytoscape. Now, Cytoscape is a tool for visualizing interaction networks. It's a very powerful tool. You can drop in a set of input values, interaction values, and target values, and then map the network in Cytoscape. So Cytoscape is a good way of visualizing networks. Intact is another interaction analysis tool from the EBI. I'm presenting multiple options because each one will have its advantages and disadvantages. We can also look at whether our genes are involved in particular pathways. Now, there's this whole pathway hypothesis in biology. We have pathways associated with things. For example, there's a cell proliferation pathway, there's a cellular senescence pathway, there's a JAK-STAT pathway, a P53 pathway. We can see whether our genes or proteins overlap with a particular pathway by dropping them into databases. So one pathway database is KEGG, which is the Kyoto Encyclopedia of Genes and Genomes. We can literally drop the gene symbols into that. It'll tell us what pathways are coming up. More powerful still is a piece of software called Reactome that looks across multiple pathways and has a very rich set of features in it. So Reactome is very powerful. We can also, if you have money, do Ingenuity, but you can use free trials. You get a free trial. A little tip, which I shouldn't really tell you, but if you have multiple Google email addresses, you just create a free trial on the email address, go for a month, generate another email address, create a free trial with the email address. And I shouldn't say that to you, but I know people who've gone for about a year doing that. And another one is metacore, which is a similar sort of approach. I suppose the only thing is if you generate 20 or 30 emails, you're going to get spam emails from Ingenuity for a whole year on every one of those email addresses, which is probably a disadvantage. Anyway, what I'm going to be talking about in the next set of lectures is about using machine learning to discover new biology and to discover drugs. That is about how we can go from biology to small molecules and how we can select biological features that are potential drug targets. So there are some databases around that. The good one is Drug Genome Interaction Database, DGIDB. What that will tell me is I can put a gene name in or protein name in to that, and it'll tell me what current drugs there are that will target that, whether they're in clinical trial, whether they're in proper use, and what indication they're used for. So this is a really good resource for potential repurposing of drugs. So say I've got a molecule of interest, say I want to find out what small molecule hits that, I put something into DGIB, I've got my data from lung cancer, suddenly I find a drug that's been used successfully in idiopathic pulmonary fibrosis. That drug can then potentially be repurposed for lung cancer, because I found this gene to be important and I found an existing drug which could have an effect in that system. So that's really powerful. What I can also use is Drug Gnome. So Drug Gnome AI is a tool produced by AstraZeneca, a drug company. And what Drug Gnome AI does is it gives me a score to tell me how druggable a particular molecule is. In biology, there are various molecules that have lots of functions. And in some cases, certain things are not very easily druggable. So transcription factors, for example, are not easily druggable. But things like G-coupled protein receptors, transmembrane molecules, anything that's kind of an enzyme is very druggable. So with Drug Gnome AI, we can actually start to drop a list of genes in, and we can say how druggable is this molecule. If we want to get a bit more detailed, we can look at something called Chembl. So Chembl is a database of chemicals. It has proteins in there, but it has thousands and thousands and thousands of small molecules. I can put a gene name in there and I can see whether there are any known drugs that target that. I can get their IC50, which is the concentration at which it would have an effect, would kill 50% of the cells. So that is important. And so I can look at that for chemical matching. And I can also look at open targets. So what Open Targets has is a database which links drugs to diseases and has a database that links drugs to genes. So Open Targets is another source for me to find out what small molecules there are out there that will target a particular piece of biology. So you can see how much value there is here. It gives us the potential to create a pipeline for in silico drug discovery. Bear in mind some of the deals that go for well-validated, well-characterized drugs are in the region of $500 million. There's extensive value here. That's the end of this bit. It's time for tea. Please feel free to ask me any questions over tea. Thank you.
they, together with Perseus, is a complete package which you can use for all your proteomic studies. So MaxQuant does analysis of the raw MS data. It takes the raw MS data and gives you the proper identification. It uses the Andromeda search engine. So you can also do the quantitation using high resolution MS data. Is everybody back? Can you ask them to be back? Garima?
So they're taking pictures with the
We don't have time for pictures. I mean, we have a dedicated time for pictures. So I have only 30 minutes. I will wrap things up. So MaxQuant gives you this opportunity to take a raw data set, and then you have these numbers. We won't delve much into Perseus, because Perseus is again something that is equivalent to MetaboAnalyst. So much of what you do on MetaboAnalyst you can also do in Perseus. What you do in Perseus you can do on MetaboAnalyst. So what kind of data sets can you run on MaxQuant, right? You can do bottoms up, shotgun and proteomics data sets. That is both DDA. And what is DDA? Data dependent acquisition. And DIA, that is data independent acquisition. So how do you work, the data sets that they work? So what kind of samples that you see? Now, if you see in the sample prep state, you can do either go for label free, you can go for MS1 labeling using SciLac, or you can kind of use iTrack and TMT. And if you see, these labels work at different levels. So label free, the primary difference that you want to maintain between runs, so you run sample 1, you run sample 2, you run sample 3, there are different runs, right? Then all the samples are not running together on the LC. Whereas if you do SciLac labeling or TMT iTrack labeling, here all your samples, if you have 12 samples, you would need a 12-plex labeling kit, and in that 12-plex labeling kit, you will run all your samples on your LC in one go, which gives it reduces the variability. But at the same time, as we had discussed earlier, it induces one more factor, and that factor is labelings are not very efficient. You lose out around 30%. So you see that both systems have their pros and cons, label-based and label-free. Label-based techniques, that means you are actually losing out approximately on 30% of the proteome just because the proteome does not take up labels. And if you're having a 6-plex system where you are running 6 different categories of labels, now just do the maths. If you lose 30% in every group over 6 groups, how much of the proteome you are missing out? You're actually missing, you might miss out more than 50 to 60% of the proteome. Whereas in label free, the problem is if your column, the LC column is not clean, if you have not run proper blanks, if your column is not properly conditioned, and somehow your nano LC pumps are not, I mean, it requires a lot of maintenance. Those who are running, I think Genal would appreciate running a mass spec requires a lot of patience as well. Sometimes they just give up. So the pistons might get loose and stuff like things happen, and the pressure drops. So if your pressure drops, just in case your buffer was not properly cleaned up, buffer was not degassed, and there was an accumulation of air in the tubing, the pressure drops. You will have an immediate impact because you cannot do something that we call the match between runs. So between run 1 and run 18, we try to match the chromatogram. We try to overlap each of the chromatograms over each other. So what is the typical? So this is again what we do. We do the sample prep. I'm not going into it. We are doing mass spectrometry, which again we discussed because it has HPLC that will give us the retention time, which is a very important component. Then you have the M by Z values, that gives us the intensity, that you also have the MS values, and then you use a tool that is known as Andromeda. It's just like the Google search engine, right? What does Andromeda do? Andromeda tells us that this is a protein database, and suppose you have given Andromeda the entire proteome database of humans, and you have told Andromeda that I'm using trypsin as the enzyme, as the protease. So Andromeda will do a in silico digestion of all the proteins, and then it will give you it in its database. It will have the theoretical M by Z values, right? So you have the practical M by Z values that the instrument is providing, and you have the theoretical M by Z values that Andromeda is providing. They match these together, and when there is a match, they identify it as a peptide, right? So you start, so this is the typical workflow. You first start with feature dissection, then you recalibrate, then you again apply a search, then you apply PSM and FDR. FDR is false detection rate, and then you do a protein group assembly, and ultimately you end. So what is feature detection? Feature detection is it's actually a big slide, so I'm just trying to matching this. So as I discussed, we have an experimental MS spectra and we have a theoretical MS spectra. So I'm trying to make things easy for you. You would have this entire PPT, you can go through it, and it's actually from the website of the people who have designed Max Quant. This is the Max Jürgen group from Max Planck Institute in Germany. So the first, you have to do a feature detection, that what is our experimental MS spectra and what is our theoretical MS spectra, and Andromeda will give it as a score. Now there is something known as decoy and target. So for every statistical study that you do, you would appreciate that there is a fair chance that we would identify or misidentify something. So what, can somebody tell me what is the difference between these two?
Extent of similarity.
There is a similarity. They look almost the same, but they are not the same. So Andromeda will have, so this is the FASTA file that you have uploaded. So I think tomorrow we can have a small demonstration of how Max Quant runs can be set up. So you upload a FASTA file, you create this in silico, and they use both target and decoy. Target means this is actual FASTA file. Then you create a decoy where you just, so if you see it's PEP TIDER, this is PIP TIDE, then you have R. So these small peptides look almost similar to each other, and they might give you almost similar M by Z ratios, but they won't, right? So Andromeda will score based on both decoy and target, right? And anything that gives a better value will be taken up. So right. So you take the MS spectra, you apply something that is centroiding, de-isotoping, and charge of transfer. I'm not getting into those details. Then you filter the top 100 peaks. So you filter the top peaks. So based on the decoy and target values, you take up the top 100. Now the FASTA file containing the protein sequence is also doing an in silico digestion. With in silico digestion, it lists out a theoretical spectra. So you have the filtered MS spectra that is coming from the instrument. It can be a thermo instrument, mostly it can also be a, and these tools, Max Quant is agnostic to the instrument that you are using. You can use a timsTOF, you can use a Orbitrap, you can use a Agilent instrument, any instrument that is creates the mass spec file most probably max quant will support it. So this is opposed to other proprietary instrumentation like Spectronaut that I think Brooker uses. Then you have other tools like Proteome Discoverer by Thermo Fisher. Those are all proprietary softwares, and you need really lakhs of rupees to buy a single license. With Max Quant, you don't need any license per se. All you need is a good PC. I think that you require for Proteome Discoverer as well. So you have a filtered MSMS spectra and a list of theoretical spectra, and beyond that, you apply this. So what is centroiding? So coming back to this, so this is the MSMS spectra. So if applicable, you will apply to centroiding, de-isotoping, and transfer to charge 1. So centroiding is, this is the initial profile, this is a centroidal profile, means you are taking only selected specific peaks, and then de-isotoping. So you remember, this, since we are using a mass spec, every molecule would have an isotope, right? And isotopes vary in what? Why? So what is the normal for carbon? C12. You have a C14 as well. So we have to do a de-isotoping because you want to remove the extra isotopes. You want to go for the mostly available isotope. But a mass spec can detect all these 3 isotopes. Sometimes when you are using a label-based method, you actually rely on isotopes to tell you, like SILAC is primarily using isotopes, heavy labeled amino acids or isotopic amino acids, to tell you between 2 different groups. Then this is the Y ion and B ion. So in the ion series, you will set these conditions between Y series, and so Y2 plus and B2 plus means charge that I had discussed in the very first morning. I think in the morning I took a fast lecture on targeted proteomics. There we were discussing that sometimes the splitting by HCD or CID leads to splitting of the peptide ions, and these peptide ions can be having charge. Rest, if it is split into something what we know as the neutral loss, that means it was, if a +3 ion was split in a way that one part carried all the 3 charges, another became neutral or it became 0, that peptide can no longer fly. Forget about flying well. Something that carries a charge 0 will not fly. So that we know as the neutral loss. So these are the neutral ions. YH2O, BH2O, it contains, if the fragment contains these specific amino acids, so these create a neutral loss for us. Specifically, there are some modification specific neutral loss if fragment contains some amino acid modification. So neutral loss is something that does not occur usually, but it occurs when you have specific amino acids and specific ions, like it can be YH2O, BH2O, YNH3, BNH3, and only in fragments that contain K, N, Q, R and DEST. Right. So one more thing that I need to understand, that you need to understand, there is too much of detail in that slide. Maybe we will have a one-day lecture on max quant, then we can go through that. One more thing about the target and decoy. You see the peptide search matrices false discovery rate, that is always based on a target decoy approach. So these are generated from target, these are generated from the decoy. So anything, so we have to set a Q value filter. If q value is 0, we will take it up as a true search. If it is something else, then we will the algorithm does not identify. So it's all about, see, nobody is seeing the peptide. We only have its spectra. We only have its m by z ratios, right? You don't see it with an eye that you can verify it. There is, and also, fact of the matter is, you are only assuming that the protein that is being synthesized is following the human genome or the mouse genome, because that's what you have put as an input data, right? Your mouse might have had a mutation in that specific area. There is no way to tell because it might have some other allele. The professor, if you remember Professor Srivastav's first talk, so the first human genome that we got was a hybrid of only 14 humans. So it does not really truly represent. So this target decoy approach prevents misidentification. So it applies, and you can apply a strong FDR, and then proteins are grouped using assembly. So this is something that I would like you to understand. So there are some unique peptides. This is something known as non-unique. Tries to identify that there are few sections of the peptide that are specific to one protein. So like these green bars are specific to protein A, but if you look at this is available in both protein A and protein B. So that gives us a score that is also known as the FDR rate. So you have some razor protein FDR. So each peptide is at most used once for identification of a protein group. Then you also have a PEP cutoff, and you also give a PEP score. So FDR is false discovery rate. So it's primarily preventing your false discovery. It's a check on mass spec, on MaxQuant. If you reduce your FDR, so we have a very stringent FDR. Now suppose you play around with the FDR and you make it very less stringent. What happens is you can identify the entire mouse proteome when you inject in a mass spectra that was generated from humans, because there would be obviously some similarity, some degree of similarity, and honestly it has happened with us. So it so happened that we were once we were doing an experiment, and I think this is with Shambhu's first data set. So we were in a hurry to get So I think this is what you have. So this is actually a SciLac experiment. Shambhu, do we have this this one? This raw data with you? Do you have it with you now? Maybe in Google Drive, right? Maybe tomorrow we can share. So you get, when you get the result, the final result that you get something like this. So MS, peptides, evidence, match features, and there are multiple features that you can play around with. Early in the morning tomorrow. Tomorrow onwards we are not having any. Tomorrow onwards we are not having any disturbance.
Deep learning, but I'm going to give deep learning, because you're going to have thousands of inputs. So immediately that becomes a complex architecture.
So remember this concept or this term architecture. It's how the nodes are connected together. The inputs leading to the hidden layer, leading to the outputs. That is called architecture. So a deep learning architecture has a complex architecture. A deep learning model is more complex than a simple machine learning model. So I'll give you some examples of deep learning to illustrate this. So a machine learning model that uses just 5 proteins to predict cancer it's going to be just a machine learning model. It's not that deep. A deep learning model would use the whole proteome, or as much of the proteome that can be measured. That can be 4,000 proteins. That will be a deep learning model. A deep learning model could use image data based upon an array of pixels and their RGB intensities to predict the presence of cancer in a pathology slide. That would be a deep learning model because it uses a matrix of maybe 2,000 by 2,000 pixels. So again, that's got to be a deep learning model because of the complexity of the data. So you see what I mean. The concept's relatively simple. Large complex networks are deep learning networks. Smaller networks are just machine learning. So essentially this is a deep learning architecture. This is just a shallow machine learning architecture. But with deep learning, we can add more functionality into our network. We can add more layers. So a shallow learning or normal machine learning algorithm might have 2, 3, 4, 5 layers max. A deep learning model could have 50 to 200 layers in it. And we can add layers into them. So what I showed you with Deep Learning 4J in Weka, you can add layers in. So you build up the architecture of your machine learning by adding layers. You can add an input layer, you can add a convolutional layer, you can add a pooling layer, you can add a recurrent layer. But you add layers in to add functionality. And those layers have this sort of functionality. So an input layer is always going to just represent the information that you're feeding in. It could be an array of pixels, it could be a mass spectrum, it could be expression array data, it could be protein array data. It's a matrix of input values. A convolutional layer, and I'll go into this in a bit more in a second, essentially takes data from the input layer and represents shapes in data. So an image, you can imagine, is a 2D array. And in that 2D array, there are going to be lines. There are going to be lines which are horizontal, vertical, diagonal, both ways. And those lines make up shapes. So what a convolutional layer is doing is essentially working its way through a matrix of RGB values, a matrix of pixels, looking for shapes, and then encoding them into the neural network. So in machine learning, we have this concept called an encoder and a decoder. And the functional layers of a deep neural network are encoder layers. They're encoding for structures in the data to try and find patterns in those structures. Obviously, if we just put single pixels in as a long, thin strip of data, there wouldn't be any association of those pixels in an image. Because you know what you're looking for is the interrelatedness of the pixels and how they exist in space. Whereas sometimes you don't need a deep neural network for proteomics data because in the first instance you're just enriching. You don't need to know if those proteins are associating with one another. You just need to represent them in the matrix to see if they're associated with a question. So we use convolutional neural networks for structures within data, But we wouldn't use them if there's no immediate structure that we're trying to find. We might just want to find, in the first instance, associations. A lot of people have tried to use convolutional neural networks for proteomic and transcriptomic data. The problem there is the false discovery rate around that is massive, because the number of potential interactions in the data that could be distant, that can't be detected by convolutional layers, but could be very real, is massive. So the data is not structured in the right way to use convolutional layers. I hate to say it, but it's usually computer scientists who don't know how biology works that put biological data into convolutional neural networks. So we have pooling layers. They merge nodes to try and reduce the dimensionality. So, if you've got a 20 megapixel image, the dimensionality of that's massive. You want to cut that down. You want to simplify it. We can do time series analysis. So we would use time series analysis for things like predicting trends in things. If you wanted to play the stock market or work out whether Bitcoin was going to rise or drop in the next 6 months or week, then you can use that. But you can also use recurrent neural networks for language processing, because language is a series of events. The words I am using are in a series. So if I say, the cat sat on the mat, then it's forming word associations in a time series. And by using a time-based neural network, like a recurrent neural network or an LSTM, we can start to model structures in language as well, to look at how words are in a particular series. So ChatGPT and things like Alexa even are using a series or effectively a time series analysis with a deep neural network. Things like self-driving cars and Teslas which drive around, they are using convolutional neural networks, but they're using effectively 4D convolutional neural networks, because they have a spatial element, but they also have a temporal element as well. So you can do a convolutional neural network in one dimension, straight line, 2 dimensions, a straight line. A matrix, which is an image, that's 2 dimensions, or 3 dimensions, which is spatial resolution, or 4 dimensions, which is time as well. So you can predict from video or layers of video whether something's going to happen or not. Then we have fully connected layers. Shallow learning uses fully connected layers, or machine learning uses fully connected layers. Deep learning uses fully connected layers. Whereas a convolutional network or a time series analysis neural network is an encoder, the deep fully connected layers are the decoder part. They're decoding patterns in that input data. So you have an encoder decoder. And that's how the algorithm works. You encode information in your data, you decode the information to try and find patterns in that encoded data. And then that fully connected layer links to an output layer, and that can represent answers to questions. It can even have multiple outputs. You could have 20 output nodes, each one representing the probability of something. The future of medicine, I'll talk about that probably tomorrow or the day after. You can imagine a situation where you take a real-time proteomic profile of the serum sample of an individual from a worn device. That feeds a signal through to an algorithm, which immediately makes a prediction of whether you are having increased risk of 20 different diseases, instantaneously, in real time, from a worn device. That's the future of medicine. But you could encode that
the challenge, as with all machine learning, is getting the data to do that and getting the device to do that. But, you know, if you have the data, you can make the algorithm. So an input layer, I'll just go into these in some more detail. I'm not going to have time for the full lecture today, but I'm going to start getting some of the concepts across. So an input layer is the interface with the world. You know, as your neural networks, you have an input layer of your eyes, your ears, your skin, your nose, your mouth. They are your input nodes, effectively. With all of those, your neural network interacts with the world. And each input node represents a feature. So it could be a pixel, it could be a protein, it could be a gene, it could be a part of a signal, a channel in the signal. It's a way of representing the world, and it is a variable. So each time point or each feature is varying through a population. Now we can transform or scale those values. What we often do is scale them to between 0 and 1 or between -1 and +1. I talked a bit about this yesterday. The reason we do that is because different things have different ranges. Now, if we looked at 20 scientific measurements, they would all have different units. Some of those things would have very large units, and some of those things would have very small ranges of units. So by scaling them, we're taking away the unit variability effect size, effectively just looking at the patterns in those inputs. We will decode them later, but for the purpose of learning and patterns, we scale them. Now, this is going to be a little bit intense, but I'm going to go through it slowly, and I might stop at the end of this so you can think about it overnight. But how do convolutional layers work? So, this is an encoder layer, and what they do is they tend to look for patterns and structures in a matrix of inputs. So we have, let's imagine a grid. Each grid has got a pixel in it, and that pixel has an intensity. We've got a big smiley face in the big center of that grid. Everything else is just plain white space. How do we represent that smiley face in that grid so that a neural network can say, this is a smiley face? Yeah? But to do that, we have to encode that smiley face within that matrix. And we encode that with a convolutional neural network. So it's basically it pre-processes matrices. This matrix can be one-dimensional, so it's effectively a line, two-dimensional, an x by y, three-dimensional, a 3D space. One way, by the way, just as a note, one way of doing drug discovery is you take a three-dimensional structure of a molecule and you represent that within the 3D convolutional neural network. And you can use that to see whether it will dock into a protein, which you also have a 3D convolutional structure for. So that's how some machine learning based docking works. So you can imagine you've got a cube, and in that cube you have a hydrogen here, you have an oxygen here, you have a nitrogen here, you have a carbon, 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 hydrogen, oxygen, carbon ring, and it's spatially located within that cube. So you're representing the 3D structure of the molecule within it's encoding. The key point here is it's encoding that structure. In an image context, this is encoding shapes within the image. So what is convolution? And this is, where we get a little bit mathematical, but convolution is essentially matrix multiplication. So all of you who were at school did maths, where you multiply this 2 by 2 grid by this 3 by 3 grid. and this is where where biologists usually groan, but matrix multiplication is the base of convolution. You're processing a grid or matrix with another matrix. So let's say matrix xi is multiplied by a kernel, which is another matrix. What does that look like? Well, this is I'll keep it in the first instance simple. So we have a matrix of numbers x1, 2, 3, 4, 5, 6 to I, x to xi. Yep. So that's just basically a one-dimensional matrix. And what we do is we want to multiply that by a kernel, k1, k2, ki. So basically we set off and start with our first position. And so x1 is multiplied by k1 and added, x2 is multiplied by k2 and added, x3 is multiplied by k3 and added. And we get an output from that called y1. So we're processing the first 3 bits of the matrix with this matrix, summing the values and putting them out to a number. So we have now processed that first part of the convolutional matrix, and we have that output, which is encoding it. What we then do is we then move along one. So then x2 multiplies by k1 goes to y2, x3 multiplies by k2 goes to y2, x4 multiplies by ki goes to y2. So we're shifting it along along the matrix. And each time we process the maths to produce an output. And that output is an encoded version of the array. Now we can put different functions in here. We can use effectively +1 multiplied by +1 multiplied by -1. We can take averaging if we want to smooth out values. But basically we can mathematically change that kernel to do different things. I'll show you some kernels in a bit. So if we were to process this, we have something called padding, which is the beginning part of the matrix, because that's not been processed through the whole kernel. But you see, if we apply this, the starting point is 0. We add some of these, so 0 times -1 is 0, 0 times 1 is 0, 0 times -1 is 0. Sorry, that's wrong. That should be a 0. The next one, oh no, we ignore the padding. The next one is 0 times -1 is 0. 0 times 1 is 0. 1 times -1, well, that's 1 times -1 is -1. But this has taken the +1 bias on it. That's why sorry, it's getting confusing myself. There's a bias of +1. That's what I forgot to add. That's why it didn't work. So 0 times -1 is 0. 0 times 1 is 0. 0 times -1 is 0. Plus a bias of +1 makes 1. Okay? We move along one. 0 times -1 is 0. 0 times -1. So 0 times 1 is 0. 1 times -1 is -1. We add a bias of +1. We get 0. So it's like going along, processing as it goes along. And so the output of this, effectively, is this encoded array, which is the input applied to the matrix. So the next step, let's look at 2D. It gets more complicated, but effectively, to just illustrate how it works, we have, let's say, a 3x3 kernel with a functional with numbers in it. We apply this grid to that matrix, and the summed value gives us here -8. Yep. We then move that along one and calculate the value.
move that along one more, calculate the value, move it along one more, calculate the value, move it along one more, calculate the value, move it along one more, and calculate the value. So that will fill these 5 squares here. We then move it down one and move it along, move it down one, along. So we're basically traversing the bigger matrix using that smaller matrix, making it move through and transforming the values. So that output matrix here is going to be used as an encoded layer in our convolutional neural network. Now, our kernels can be various sizes. We can have a 3x3, a 4x4, a 5x5. The bigger the kernel, the longer it'll take, but potentially the more detailed and accurate the encoding we get. Because if we can represent trends over bigger areas if we use a bigger kernel. What sort of kernels can we use? Well, if we look at these, this is a box blur, so this just takes everything multiplied by 1, but then divides by 9. So that's effectively averaging across that matrix. The matrix goes to a single point. It's in effect lowers the resolution and blurs the image. That might be just a way if you want to just kind of remove noise. There might be single pixels that by using that are causing problems. You would remove those by taking that blur. You can do the same thing. That's called a Gaussian blur. You're kind of biasing it a little bit more by using a different value, and the numbers are different. This one is called a Sobel edge detection. Sobel edge detection is used to determine edges, and it works to some extent to find horizontal and vertical edges. But for encoding, sometimes you want to represent shapes, and to represent shapes, you want to detect edges. So in your smiley face, there were going to be edges around the circle, and maybe edges around the eyes and the smile. You want to detect those edges as shapes. So that's the simplest possible form of doing that. So you're using one matrix to detect edges. But you can have multiple different parts of the convolutional neural network all applied to the matrix in parallel. So you could have layer 1, which is a horizontal line detector. So that's reinforcing horizontal lines. Everything else is played down. You have a vertical line detection. You have a diagonal line detection and an opposite diagonal line detection. So each one of those is basically 4 kernels that are applied to 4 parts of the matrix separately but in parallel. And then you combine those into one set of results, which represent an increase in the size of the matrix but different line states. You could pool them as well if you wanted to. So what this is doing is going through images, detecting things and encoding things. Just to illustrate what the layer output looks like, this is what a convolutional output without going through processing of something might look like. So each pixel has effectively detected a different edge. Some of them horizontal, some of them are just transitions, some of them vertical, some of them diagonal. Here we've got circular lines, arcs, angles. These have essentially been encoded into the convolutional layer of the neural network. And that information can be used in a classifier. So, any questions around convolutional neural networks? It's a whole subject. You could probably do a degree in convolutional neural networks. There's that many things to consider, and it takes
They get very complicated. As I say, can be in 200 layers. And strategies are there that will cope with that much information. So the next layer is pooling layers. How are we doing for time? A pooling layer merges multiple layers to simplify the network. We could end up by having a matrix of 20,000 by 20,000 features. A pooling layer basically cuts that down by summing or averaging. You can take one way of doing it is you work across the matrix. So you work your way through. You're looking at a 9 by 9 grid. You just take the max in the 9 by 9 grid for that. Max for the next one, max for the next one. So you have a smaller array of max values, but you don't overlap. You just literally go through the grid. So you take your first 9, you move along 3, and take the second 9. You move along 3, take the third 9. So by doing that, you're pooling, but it's not a rolling pool. It's literally summing those 9 pixels up, but they're not without overlap. I think probably I'm not going to go into time series neural networks today, but now have a look at convolutional neural networks. Have a look at the basis behind them. It's quite a complex topic. Again, most people use them without fully understanding how they work. They have the broad principles, but if you ask them to derive a convolutional neural network mathematically, they would be very stuck. And so having an understanding of what they're used for, having an understanding broadly of how they work, is an advantage. Any questions? No? The convolutional neural networks piece is a bit of a simplification. It is a complex topic, but hopefully gives you a flavour of how they work. Okay, thank you very much.